So let me share my screen first, and then we'll be starting with the session. So is the PowerPoint presentation visible to you all? Can you please put it in the chat box so that I'll know if the PowerPoint is visible to you? Can you please respond? Is any one of you able to unmute and speak?
Yes, ma'am. Visible.
Okay, okay. Okay, so good evening to everyone present here, and I am Guru Priya, Prime Minister's Research Fellow in the Department of Civil Engineering, IIT Madras. So this is a live practice session for the NPTEL course Applied Environmental Microbiology. I'll be handling all the live sessions for this course for all the weeks. This will be our first session. Um, Here, basically, what we do is we try to solve some questions related to the this week's content. The content that was posted for week one study. So the basic idea of this session is like to give you a practice and to solve your doubts if you have any. So you are free to ask any doubts and any questions. And The whole module will be something like I'll give you practice questions for which you will give me the answers, and we'll be discussing the answers. So this is how the session is planned. uh, Basically, I'll not be doing any teaching or any seminar session kind of a thing. It will be a very interactive session. So I hope all of you uh, will interact and will gain good knowledge out of this. Okay, so we'll start. To start with, in the applied environmental microbiology course, the first week had the following contents. Uh, the topics on the history of environmental microbiology, microscopy, cell cytoplasmic membrane transport, DNA, genes, genetic elements, central dogma, all of this was covered. And so, why do you think? I'll start with a very basic question just to have a little bit of interaction with you. Why do you think the microscopy is very important in microbiology?
Because we through our naked eyes we can't see the microscopic element, just like bacteria, virus, archaebacteria, etc. So microscope would be the most important thing in the field of microbiology, to see all the mechanism, all the activities in microbial world.
Great, great. So that's a very good answer. Um, So you have a basic understanding of what microbiology is and how microscopy is used to like study in the field of microbiology. Before
Yes,
start,
ma'am.
yeah, okay. Thank you. Thank you for your response. And before starting with the question and answer session for today, let me just tell you that you people are from varied backgrounds. Like you may be a biotech person, or you may be a civil engineer, you may be an environmental engineer. And I also don't know if you people belong to the UG or PG categories, basically with your studies. So this session, I have planned it to be a simple one, uh, just for introduction and practice to get to know how the sessions will move on. If you have some suggestions on how the session should proceed, you can tell me at the end of the session. Okay, so in between, if you have any doubts, uh, I would prefer you put it in the chat box so that it doesn't disturb the session. So, yeah, coming to the first question. Um, how we are planning to move is that I'll give you a few minutes. You can read the questions and post the answers in the chat box, and then we'll discuss about it.
DNA is bound with histones.
Okay, yeah. Okay. I would request you people to please put it in the chat box so that I can see answers from many of you. Okay. So as most of you have told, let's see the answer. It is C. Um, there are a few more questions that will go with this topic. So I'll, for now, proceed with the next question. Thank you, Shivani and Ruchira. You can just read the question once more. Okay, great. I guess this is a pretty easy question, but yeah, only Nidhi Praveen has got the answer right. It's both fungi and protozoa. So the question clearly asks which of the following is or are not a prokaryote. So it can have multiple answers. Okay, all of you are partially right.
Yes, ma'am.
Okay. So now we'll proceed with the third question also, and then we'll have a small discussion in the topic. Great. So this question was pretty well answered by all of you, right? And it's mitochondria. So do you see any uniqueness or similarity between these three questions? What was common with all these three questions? What topic was common? And we'll have a discussion on that topic only now.
Genetic material.
Okay, right. Nidhi, you are perfectly right. So the discussion is about eukaryotes and prokaryotes. I guess all of you are now familiar with the term eukaryotes and prokaryotes. Um, just a minute. I'm sorry. Um, I guess all of you are now familiar with the term eukaryotes and prokaryotes, right? So we have to first know what is the basic difference between a eukaryote and prokaryote while studying microbiology. Because at the end of the day, most of them, all of them are going to be single cells. So what is that difference between a single cell prokaryote and a single cell eukaryote? There are multiple differences as listed here. The main difference is that prokaryotes do not have a membrane bound nucleus. So their new genetic material is just embedded in the cell. Do you know what that portion of the genetic material embedded in a prokaryotic cell is called? Any of you would
Ma'am,
like to
can
try?
you please
Yeah.
repeat once again? Ma'am, can you please repeat once again?
Okay. So we all know that prokaryotes do not have a membrane-bound nucleus, right? So, but their genetic material is embedded somewhere in the prokaryotic cell. Do you people know what that uh, particular portion is called, or that segment of the cell is called?
Nucleoid. Nucleoid.
Okay,
Ma'am, it is nucleoid.
nucleoid,
Nucleoid.
right? So that is a nucleoid. And when it comes to the eukaryotic cell, that is where we are going to call that portion as a nucleus. Eukaryote means means true, and karyotic is nucleus. So the organisms that contain true nucleus are called eukaryotic. And the other differences is generally prokaryotic cells are like smaller in size compared to the eukaryotes, and the prokaryotes lack membrane enclosed organelles. Membrane enclosed organelles include lysosomes, Golgi complex, mitochondria, chloroplasts, etc. All these organelles are not present in the prokaryotes. And as listed here, we can see many differences. And as asked in the question, one of the key differences is that uh, the chromosomes of prokaryotes lack histones. Anyone has an idea about what histones are? What are their functions in the cell?
Actually, histone is a protein and which packed the DNA. I mean, DNA coiled with the histone. Uh,
Right. You're absolutely right. So I guess people who are from a biotechnology background or microbiology background would obviously know what a histone is. So let me explain it to the people who don't know what a histone is. Histone is basically a protein, as told by Nikita and the person who answered. I'm sorry, I will not be able to see a name. So histone is basically a protein. It helps to like coil the DNA so that we can pack it inside a nucleus. Have you ever wondered how such a long DNA strand is just packed inside our cells? It is because they are wound over the histone protein, and hence they are comfortably being packed inside the cell. And Also, there are many functional differences between a prokaryotic and eukaryotic cell. So the last question, or the third question that we dealt with, was basically on mitochondria. So when coming to mitochondria, mitochondria, as you all know, is the powerhouse of the cell. So it's going to give ATP molecules, that is adenosine triphosphate molecules, which are rich in energy. So when you are going to break the ATP molecule, the cell will yield the energy. In case of prokaryotes, the mitochondria is absent, right? So how does this energy generation process take place in a prokaryote? Okay, I guess all of you are silent for this question. Let me explain. So mitochondria basically has few proteins embedded on its membrane where the electron transport take place. So next week we'll be seeing about metabolism or the energetics, right? So that time maybe we can also cover this topic. Now I'll just tell you a brief that mitochondria has many proteins in its membrane, and these membrane will be present in the plasma membrane of the prokaryotes where the electron transfer will happen. Okay, so there is no need of a specialized mitochondria in a prokaryote to carry out this energy generation function. Okay, so this is a basic understanding of what a prokaryote and eukaryote is. You can read multiple sources and you can understand vast differences between them. And when you're going to ponder more on this topic, if any one of you have any doubt, you can just post it in the chat box so that I can clear it out regarding this prokaryote and eukaryote topic. In the meanwhile, let me show you this clear differentiation in a pictorial format between a prokaryotic and a eukaryotic cell. So you can see a prokaryotic cell has this genetic material here, and this space, as we discussed, is called a nucleoid. They have a flagellum which is used for locomotion. They have few inclusion bodies which acts as storage. Inclusion bodies can be made of many molecules, like different molecules. They act as reserves basically. And this is the chromosome genetic material. You can see these small projections from the cell, which are called fimbriae. They help in the attachment. And another important characteristics of prokaryote is this capsule. Uh, Here the capsule is the outer covering of the prokaryotic cell. Again, they have ribosomes, for which are the protein machinery of the cell. They have a cell wall and the plasma membrane. So to be clear, this is a typical prokaryotic cell. Few prokaryotes may contain flagellum and they may have differences in their flagellum. That all depends on the genus, species, and we are all aware of the vast diversities among organisms. Uh, so here, this is just a basic overview of how a prokaryotic cell can look. And when we come to the eukaryotic cell, I have an example of both fungal and a protozoan cell as asked in the second question. So here you can see a very developed nucleus with a nuclear membrane and the membrane bound organelles like mitochondria. And you can see their plasma membrane has all these components and stuff. So if you're interested, you can ponder more about what each of these organelles can function and how basically they help in the survival of the organism. Okay, I see no questions in the chat box now. I guess the topic is clear now. Let me just move to another set of questions so that Okay. Now you people can think and answer for this. Okay, I got two responses which tells B. Right. So as you people have told, it was Carl Woese who described the three domain classification. And can any of you tell me what are the three domains that was described by Carl Woese? Okay, I don't see any responses in the chat box as well. Right, Bensi. That's bacteria,
Archibald,
archaea, and eukarya.
yes.
Oh, so yes. Now we go to the next question. I'll just wait for a few more responses. Okay, I see a lot of B in the answer. I guess the question is pretty easy, but a bit tricky, right? Okay. I would request you to see the last option, which is obviously the answer, which has both A and B. I got a multiple response which says A, multiple responses which say B, but the answer is both A and B because extremophiles are organisms that can survive in extreme conditions. So, be it psychrophiles, be it alkaliphiles, be it acidogens, they are going to survive in extreme conditions, extreme things. So, barophiles basically love high pressure conditions. Extremophiles basically love extreme conditions. So, high pressure conditions can also come under uh, extreme conditions. So, both A and B will be answer for this question. Just look at all of the options, think for a minute, and then answer. Okay, so that you don't miss on key things. Okay, this is a pretty easy question. You just have to tell me if it's true or false. Okay, as you have heard in the okay, if you have watched all the lecture videos, you would have obviously known the answer for this question, even if you don't have any idea about how individual genes work and stuff. It's just pretty simple that it's true. Yeah, MCRA gene is linked for methane production in methanogens. So now let's have a little bit of discussion. Before that, why is this three domain classification important? Can any of you answer for this? What is the significance of this three domain classification? That's okay if it is right or wrong, it doesn't matter.
Classification
We
must
just
be
have to know.
Ma'am, classification must be needed for their differentiation between their structural and functional uh, composition.
Okay. And here, like, what exactly makes us differentiate these three? Okay, let me explain. So, as we saw in the previous topic, what we saw it was about prokaryotes and eukaryotes, right? So, here we can see we have three domain classifications in which one of it is eukarya, which is basically all eukaryotes will be grouped under this eukarya category. So, the other two are going to be prokaryotes. The reason why we have two domains in the prokaryotes is that there are major, as the person who answered told, there are major structural and functional differences between both of them. You can see the archaea contains extreme halophiles, hyperthermophiles, and methanogens. Uh, One of the very evident similarity is that these bacteria are extreme condition loving. Sorry, these organisms are extreme conditions loving organisms, right? Uh, but whereas here we have variety of microbes like gram negative bacteria, gram positive bacteria, etc., which are pertaining to normal conditions or mesophilic conditions. Okay, so what makes them withstand this extreme conditions? I mean, what makes archaea withstand this extreme condition is that their biomolecules are the structure out of which they are made up of. So they have a few biochemical advantages which makes enables them to survive in this extreme condition. So that is the key difference between this bacteria and archaea. And based on this only, we have this two domains in the prokaryote category and a very distinct domain called eukarya. Okay, so here maybe if you watch this image on the top very keenly, you can see that uh, bacteria contains mitochondria and chloroplasts. So this is a bit contradictory for what we have been discussing so long, right? We have seen mitochondria and chloroplasts as uh, organelles of a cell and not some organism kind of a thing. So you can see that a red arrow follows here and a green arrow follows here, which ends up in the eukarya. Um, this is just an information for you to go and ponder upon something called endosymbiotic theory. I'll just give a gist of what this theory is. It basically states that mitochondria and chloroplast once existed as individual organisms and they were incorporated in a cell, uh, becoming their photosynthetic unit and the energy machinery, and they were very helpful for the survival of the organism that due to evolution they just stayed as an organelle with that organism, and hence came the origin of eukaryotes. So this is what endosymbiotic theory states. If you are interested, you can just go and read upon it. um, with this theory is basically very important when we talk about this classification or how eukaryotes domain got built up, and this is a very important theory in that aspect. So having said that, can you tell me uh, a very basic reason with which you can tell mitochondria and chloroplast might have existed as individual organism once upon a time, very long ago? Does anything come to your mind with which you can say, okay, this is a reason why mitochondria and chloroplast can be could have existed as individual organisms? Right, Nidhi Praveen, you are exactly right. Both mitochondria and chloroplasts have genetic material. So as we have discussed in one of the previous question, mitochondria's distinguished feature is that it has a genetic material. Similarly, chloroplasts do also have genetic material apart from nucleus of the cell. Okay, coming to the basic differences between these three domains, as you can see, this is sulfolobus archaea, and here you see E. coli. Maybe most of you would have heard this name, Escherichia coli, be it any background you would have heard Escherichia coli. And here is amoeba. Amoeba also, maybe people who are from science background in their schooling would have obviously heard about amoeba. So that is an example of eukaryote. The basic difference is that, as we all know, archaea and bacteria fall under this prokaryotic category, whereas eukarya are eukaryotic, obviously. And one of the key differences, as discussed in the lecture also, is that there is variation in their cell wall composition, and the archaeal membrane does not contain any peptidoglycan. The bacterial membrane, on the other hand, contains peptidoglycan, and the eukaryotes have a varied composition in their cell wall. So when you talk about microbiology, you are confined to amoeba, fungi, protozoa. I would also like to remind that plant and the animal kingdom also come under the eukaryotic category. So you can imagine the vast difference in the cell wall. You can imagine our cell wall, the human cell. If at all any animals have a cell wall, you can imagine the vast diversity in it. Okay. Did I speak anything contradictory a couple of minutes ago, or was everything fine with whatever I was telling? I'm just trying to interact with you and know whether you people are interested or. Okay. Okay, we'll get back to the topic that there is a vast diversity in the cell wall of eukaryotes, and as told, the one contradictory statement that I was stating was about the cell wall of animals, wherein animals do not contain any cell wall. In the plant cells, you can see it is made up of cellulose. Okay, we'll move to the next point now. That is about the membrane lipids. I, as discussed in the lecture, you all know that the archaeal membranes usually have ether linkage, whereas the bacteria has the ester linkage in their membrane. And also one more uh, key difference is that the bacteria usually has formyl methionine on as its first amino acid during the protein synthesis. So this protein synthesis, I hope you have all come across the central dogma. So what is protein synthesis called? What is the step of protein synthesis in any organism called?
In translation,
Right, translation. So that in translation, as we all know, we have triplet codons, which each codes for a particular amino acid, and in that we will have start codons. And then the first amino acid that uh, comes in the translation process is usually methionine in archaea and eukarya, whereas it is formyl methionine in bacteria. This is a very key difference between these organisms. And as we have discussed, you can see that few features of the archaea uh, can match with bacteria, but it mostly follows the eukarya pattern. So that is why this archaea lies in the middle of this three domain classification. So there is a reason why this diagram should be drawn only in this way, and not, you cannot invert archaea here and bacteria here, leaving eukarya aside, or you can bring eukarya to the middle. That will not work. Okay. The main reason is that archaea has contributing features with similarities from both bacteria and eukarya, but it is more similar to eukarya than bacteria. You can see from the branching of this diagram. So that is about the basic understanding of the three domain classification given by Carl Booth. I guess first we saw about prokaryotes and eukaryotes, and gradually we moved towards speaking about specific organisms. Now I will give you a couple of minutes to answer this question so that we'll solve a few more questions and go for another discussion. Obviously, you would have figured out now this is going to go to microscopy and other things. You can try answering this question. Okay, right, right. I've got good number of responses, and yeah, it is true. Okay, now I'll give you enough time to just go through the question, see which of these answers is correct. Okay, as most of you have answered right, the first option is right. The differential interference contrast microscopy basically works based on the refractive index difference. It images based on the refractive index difference, and the atomic force microscopy does the topography mapping. What is a topography mapping? It is when you have a surface. This atomic force microscopy usually uses some kind of a force. Uh, it just vibrates over the surface and it can map the crests and troughs in the particular surface, and finally it can show us what's the topography of, of the surface that we are testing. And the confocal scanning laser microscope, basically the samples usually contain fluorophores, and when you are going to pass laser light through it and collect the reflected light, you are going to get the image based on the fluorophores and the structures to which these fluorophores are attached. And the electron microscope, that is pretty easy. That the scanning and we have two methods called the scanning and transmission electron microscopes. uh, they basically vary in their mechanism, where as the name suggests, the scanning electron microscope just scans the surface that is under examination, whereas the transmission electron microscope penetrates. uh, I'll just explain more about this in the discussion part. Yeah, here here we have, as we discussed, you can see that the differential interference contrast is imaging based on the differences in refractive index. So you can clearly see how the structure looks. And here you can see in the electron microscope, uh, basically it is electron microscopy only, but this image was imaged using the TEM, that is a transmission electron microscope, and this using scanning electron microscope. So now you can see what is the difference. When you are going to just scan the surface, you're going to get the information about the surface only, and when you're going to allow it to transmit through, you can see what information that you can get in the results, right? And in the confocal microscopy, basically you get this color differences also. And in any other microscopy per se, you will not get any colored images. So this electron microscopic images are also like kind of a black and white images only. You can use softwares and then add these colors and contrast to the structure, but the raw data that you receive from the microscope is not going to be colored. And finally, the atomic force microscopy. You can see this is very different from all rest of the images, where it doesn't show any complete structure or something. It shows how the surface just looks. You can see that this is the perfringolysin O toxin from the Clostridium perfringens organism, right? So you can see how the protein or toxin basically looks. It has a surface structure similar to this. So as we discussed, this confocal microscopy, this uses a single photon to illuminate one plane of a specimen at a time, and that is why you tend to get such images which can also be 3D images. And the differential contrast, as you can see, it ultimately provides 3D images. So when you are asked the question about 3D images and given both this differential interference contrast as well as confocal, uh, you would most preferably choose DIC because it exclusively provides 3D images. So as told, this atomic force microscopy uses a metal and diamond probe. As you can imagine, maybe like something like a stick with some triangle attached to its end. That is how a probe of atomic force microscope is going to be. And you're going to scan that probe through the surface, where you will get an image as shown here. And the electron microscope per se, you can see that as asked how much it can magnify, how much the resolution can be. Maybe if some of you are not clear with the term magnification and resolution of a microscope, magnification, as the name suggests, it's the ability to enlarge the image, and resolution is another different thing, which means that how far you can separate two different points distinctly. That is, when you're going to image, if you're going to have 2 points, and at some resolution, you will not be able to distinguish between both will appear somewhat together or overlapped. So in that situation, that is not called the resolution. That distance between or that thing that you're using is not called resolution. When you're able to distinctly separate between those 2 points, how much of a distance you can separate them, that distance is known as the resolution of that microscope. These two are very important properties when you're going to study about a microscope. And based on what is your need, um, or what purpose the image is gonna help you, you can choose either of this. And there are also a lot more microscopic techniques, so you can choose based on what you will be uh, needing ultimately from the image analysis. So now, since we have seen some basic things about the cells and microscopy and stuff, we'll see about something else also today, that is about the gram positive and gram negative bacterium and other things. I'll wait for a few minutes for you to read the question and choose the right option. Okay, there are two responses as of now. Okay, a few answers for B and one for A. Okay, all of it is B now. Okay, before seeing um, the answer for this question, do you have any idea about what a gram positive and a gram negative bacteria is? Maybe any of you can unmute and answer because it'll be pretty long to type.
Gram positive bacteria, ma'am, they are taking the stain easily, and gram negative, they are generally not taking the stain easily because of periplasmic extra layer.
Right, right. Okay, okay. So the answer is B, as most all of you have answered rightly. We will have a discussion about this now. So here you can clearly see uh, what is a gram negative cell wall and a gram positive cell wall. Before going to that, what is even called the gram thing? So gram is Hans Christian Gram. He is a scientist who discovered this feature, and hence the bacteria can be termed as a gram positive one or a gram negative one. As told by one of you, um, what we do is we take a microscopic culture, or we take a microbial culture and add the crystal violet. I hope you have heard about the crystal violet stain. So we add it. We also add iodine as a mordant. So the basically the role of the iodine here is not to impart any color. It just makes a strong attachment between the cell and the crystal violet type. And then you do ethanol wash, basically ethyl alcohol wash. So when you're going to wash with ethanol, what happens is that only few cells will be able to retain the violet color which was imparted, and the remaining cells are going to give away the violet color. Basically it's going to be washed off. So after those are washed off, what you can do is you can add saffronin, which is again a dye, and this saffronin will be coated on, or will be bound to the surface of the organisms which are now colorless. Whichever organisms that are retaining the violet color or the purple color are termed as a gram positive one, and whichever are going to give up the color and take up saffronin are going to be called as gram negative bacteria. Okay, so this is the basic procedure for gram staining. Whenever you use a dye to visualize the organisms with the help of a microscope, you're going to call it a staining technique. That is a very important technique in microbiology. And using the staining technique, what you do is you just differentiate the microorganisms either with respect to the background. Sometimes you just add only one dye, like like you have added just to see them, because when you see it with a white background, again if the cells are going to be white, you can distinctly see. So you will just use, sometimes you will use it for very unique purposes, like you have acid stains, you have basic stains, and you have many staining techniques also. So here, since gram positive and gram negative term you're going to be hearing multiple times in microbiology, you should be aware of what gram staining is, and this is the procedure. Okay, now we have discussed what gram staining is and stuff. What makes this crystal violet to stay in certain bacteria, and why it is washed off in some and saffron is taken up? So that is what is the key feature. So here, as you can see, the gram positive cell wall usually has this plasma membrane, a cell wall. So you have this peptidoglycan layer, And you can also see teichoic acid here. Whereas in the gram negative cell wall, you can see that there is a plasma membrane. You can see there is a space in between, which is known as the periplasmic space or the periplasm. And finally, you have another outer layer, which is called as the lipopolysaccharide layer or the LPS layer as generally termed. It is called the lipopolysaccharide. So with the basic biochemistry understanding, you know that lipo stands for fats, polysaccharide stands for sugar moieties, where they are in polymeric form. So lipopolysaccharides is basically made of lipids and sugars. You can see that they have these three components in them. And these lipopolysaccharides are present outside the cell wall layer. Okay. So this is the visual difference that you can see from this image. And the characteristic difference that we can infer from this table is that the peptidoglycan layer is thick in the gram-positive bacteria. You can see here, this is the peptidoglycan layer and it is visibly very thick, represented very thick. Whereas here you can see that the peptidoglycan layer, that is the brown color or the brown and pink rod kind of a structure that is present, is very thin. Visually you can see two layers or three layers of it here, but you can only see one layer of it here. So that is what this thick and thin basically means. And teichoic acid is present in many of the gram positive, whereas it is absent in the gram negative thing. And the periplasmic space is absent in the gram positive. You can see you have a plasma membrane, you have a cell wall, that's it in the gram positive. But whereas here you have this space, you have an outer layer, and the space between the outer layer and the plasma membrane is going to be called as a periplasmic space. Lipopolysaccharides are almost completely absent in the gram positive cells. And these other differences that you can better know for your knowledge is that the flagella structure, or when the flagella is going to be attached to a gram positive or a gram negative bacteria, they will have a different arrangement in the cell wall. You all know that the flagella will be attached to the outer layer of the microbial cell. So because there is difference in the layering of these outer layers, that is a plasma membrane or cell wall or the lipopolysaccharide layer between the gram positive and gram negative, the arrangement of the flagella is also going to vary. And the last 2 points that you can see is the resistance to physical disruption. Physical disruption meaning when you are going to just subject it to some mechanical methods or grinding or something, the gram positive is going to resist highly, whereas the gram negative will have a lower resistance to physical disruption. Similarly with enzymes also, lysozyme is an enzyme that can disrupt this cell wall structure. So when you are going to see in a gram positive cell, your cell wall disruption is going to be high in the gram positive, whereas in the negative you can see that it can be very low. So now coming to the final conclusion of this topic is that when crystal violet is added in the gram positive, it now you know that it might get attached to the peptidoglycan layer only. But here you don't have the access to the peptidoglycan layer. So it initially has this, gets the stain, the gram negative initially gets the stain, but when you are going to add ethyl alcohol, it is going to be washed off from the due to the lipopolysaccharide layer. And finally it takes up the saffron stain and shows a pink color. Whenever you're going to study about any bacteria, one of the first questions that you will end up asking is whether it is a gram negative or gram positive. So you will be performing this gram staining with whatever bacteria you're going to handle. I hope this was clear. And if you have any more doubts, I'll give you one more minute so that you can either unmute and ask, or you can put it in the chat box. Or let me know if it's clear. We'll move on to the next topic. So, shall I move on to the next question? Okay, I'll take it as yes, and I'll now show you the next question. Okay, this is again a pretty easy question, and if you have just gone through the lecture videos, you will be able to answer this. Okay, and I'm a bit sorry for the last option that's given here. It should be both A and B, and the C will be none of the above. So it cannot be all of the above. I'm sorry for that. You can anyways post your answers, considering the last option to be both A and B. Okay, most of you have answered, I mean, D, and one person A. Can you just give a few word or a line explanation of why you told the answer in a chat box? Or maybe what is the metabolic advantage? Just a 2 3 word answer would do. Anyone in the chat box? I'm just waiting for your response so that we can move on with the answers. Okay, right. If any of you would like to unmute and answer. Yeah, okay, Ruchira. Relate with the external environment, right? Okay, I'll move on with the answer for this question. It's basically both A and B, high surface area and lesser volume. So how does high surface area and lesser volume will be metabolically advantageous is generally with metabolism we consider something called a surface to volume ratio. Surface to volume ratio usually considering cells to be spherical. In the lecture video you would have seen formula calculation on how you calculate this surface area to volume ratio. When this is going to be higher, you can expect very good nutrient uptake as well as excretion from the microbial organism. So we all have heard about maybe nanoparticles. Nanoparticles are used in many fields. The main advantage why we are going for nano sized particle is because they have a high surface area. So as the size of this particle is reducing, the surface area is going to increase. And the high surface area is kind of very helpful thing in uh, many things. Maybe when you use nanoparticles in medicine or like drug delivery or something, or in other applications, these nanoparticles are of high advantage. Similarly, because their surface interaction is going to be better. Similarly, when you're going to talk about microbes, when they have higher surface area, obviously in a fraction, when the numerator is higher, that is the surface area is higher, you're going to have a major advantage. And the advantage in very simple terms is that it can easily interact with the environment. It need not struggle, and the environment and the microbial transfer or exchange of molecules is going to be easy. uh, ex- you all know that microbes have a very complex yet simple life, though. So metabolically the reactions are going to be a bit complex. Yeah, biochemical reactions have its own complexity, but at the end of the day, this molecule transfer is going to happen very swiftly or easily when you're going to have a higher surface area and a lesser volume. So this all comes with the kinetics of the molecular transport between the microbe and the environment. Okay, that was one question which I felt important in this introduction topic, so I added that, and the discussion about that is over now. Now we'll move on to the central dogma part, which is pretty interesting. And yeah, I'll give you a couple of minutes to answer this question. Okay, 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 I finally got one B. Let me ask Nidhi Praveen, like, why is it B? Because I have got mostly A, and can you state why it is B so that we'll see what's the reason? Exactly. So that is the nuance in this question. So you should be pretty careful with all the terms that are given in the sentence. Okay. Start codon is associated with the protein synthesis. Yeah, true. But the start codon usually binds to what? It binds to the mRNA, right? We are talking about translation here. In the transcription part itself, the DNA has been converted or been transcribed to mRNA. And now we just have a mRNA template to which the ribosomes will come and bind, and the start codon or the tRNA is going to come and bind, right? So since we will be talking about RNA in the protein synthesis, this statement is false, right? Yeah, we'll discuss more about central dogma, and before that, I'll give you a few minutes to answer this question. Right, I've got three responses. And based on the responses, yeah, it is transposons. So to explain about this, transposons are generally also, do you know the common name with which we call the transposons? That's not very scientific. I'm just asking for a common name so that you can relate to this when you see such a statement. Okay. Transposons are also known as jumping genes. So I think now you can better relate with the question than before. So transposons are also known as jumping genes because they are small segments of DNA that can move from one region of a DNA molecule to another. So when you have a double helix or the double stranded DNA, this jumping genes are a few base pairs or a part of a gene that can uh, move from one position and get attached to another position in the same DNA, and that is why they are also known as jumping genes. Okay, so this is a pretty interesting phenomenon uh, in the field of microbiology. Yeah, um, so if you want, you can read more about this, and yeah, sure, it's pretty interesting. So coming to the other options here, chromosome, we all know that chromosomes are not small segments of DNA, so therefore they are not going to fit in this question here. Uh, Plasmids, what are plasmids? Do you have any idea of what are plasmids? Can anyone tell or put it in the chat box? Like transposons are referred as jumping genes. What are plasmids? Okay, I'll wait for a one or two responses more. Okay. Right, finally. So all of you have this term extra. Yeah, it is right. It is not extracellular. Obviously, it is going to be present inside the cell, so it is not extracellular. They are extra chromosomal genetic material, which means that apart from chromosome, there is going to be one more DNA molecule. So that gives the name extra chromosomal genetic material. So these plasmids are again DNA molecules which do not fall under this chromosome category. They are a very distinct and a very separate entity that are going to stay in few of the microbial cells. So even the DNA strands are going to have some genes which codes for few functionalities, and these functionalities will most probably not be related to the essential survival of the microorganism. So when you know about this chromosome thing, the genes in this are going to code for important proteins. Like if you know, glycolysis is one important metabolic activity that we have in our cell, and that requires quite a lot of enzymes. And Microorganisms also have something called this operon concept. So genes are very important, and we all know that genes, this DNA is going to transcribe to mRNA, and which is going to translate to protein. So DNA is very essential for this protein synthesis. But are these proteins very essential for our survival? When it comes to that topic, the proteins that are coded from this chromosome are said to be very essential for survival. If that is going to be defective, it is either going to cause any, it is because of any mutation, so you are going to lack that protein and end up with something very something that is not very ordinary, and maybe the cell can also die because some essential protein is lacking. Whereas in the case of plasmid will also have genes that code for some useful purpose. It may not be ultimately essential for the survival. So that is why they are called extra chromosomal genetic material. And these plasmids can also be like, what to tell, transferred from one cell to another. And this Pili, if any of you would have heard about it, you would have known that pili is a tube kind of a thing where when two microorganisms have to interact for their genetic material transfer, so this pili uh, comes into picture. The tube that connects the two microbes is called pili. Let me be clear again. In these options, the transposon also refers to a part of the DNA, so which are also known as jumping genes. Uh, Whereas plasmids are extra chromosomal DNA. This pili do not even fall under any of these DNA or chromosome category. They are not nucleotides or nucleic acid molecules. Okay, so this pili is just the connection between two individual microbial organisms, like one cell and other cell. They have to have a genetic material transfer between them, one strand transfer between them, then this pili comes into picture. I hope this question was explained clearly. So let me move on to the next question with regards to this DNA and the central dogma. And before that, if any of you are not aware of what a purine and a pyrimidine is, you can also let me know in the chat box. We'll anyways going to discuss about it. Right. I have got a pretty good number of response now. So we'll just see the answer for this question. Okay. Almost all of you have answered it C only. So it is adenine and thymine are purine. Before explaining the answer for this question, we'll just solve one more question, and then finally we'll look about this DNA and the entire process of the central dogma. You can take a couple of minutes extra and just read the options carefully and give the answer. And if any of you find any of these terms very new, and or you're coming across these terms for the first time, just wait for a couple of minutes. Once I get few responses, we'll just straightly go into the discussion for this topic. Okay, there is one response. Okay, now I'll just give a pretty little reminder to read the question carefully and then read the options carefully and answer. I'll just wait for a couple of minutes more. Right, right. There are multiple answers for this question. So if any of you would like to try, yeah. Okay, great. We'll move on with the um, discussion part. And before that, okay, D option. Okay, just a few minutes back, I told you in the previous questions that tRNA helps in the transcription process. While I was telling about the process, uh, I'm just curious to know what is wrong in this B option. Just type which is the word that is wrong in the B option in the chat box. I guess Nidhi Praveen and Ruchira Das are the persons who have told it's both B and D. Okay, what is the reason that B is wrong? Right. So yes, as these people have told, B and D are the false statements. Let me just give a brief overview about all these four options, and then I'll explain the topic uh, in a continuous manner. So here the first one talks about nucleic acids attachment. We'll be seeing in the pictures how a nucleoside attached to a phosphate group is called a nucleotide, what a nucleoside is, and why the statement is true. Here, as Nidhi Praveen has mentioned, the wrong word in this is parallel. I'll just show you a picture of a double helix of a DNA, and we'll figure out why this it is not parallel. So you all have understanding that there are going to be two strands which are which, which are helically going to be connected. That is what we call a double helix of a DNA. As you see, one helix, there it's going to be in parallel direction. It is going to seemingly be parallel. Why are we not uh, telling it as a true statement? That we'll just see in a couple of minutes. Here, coming to this C statement, protein is not a polynucleotide. Yeah, protein is not a polynucleotide, but instead it is a polypeptide. Poly, as you all know, stands for multiple units. So protein is made up of polypeptide units and not polynucleotide units. Okay. DNA, maybe you can call it as a polynucleotide, but not a protein. tRNA helps in the translation process, as I told in few slides back. It doesn't help in the transcription process. So now we'll see this slide. Okay, we'll start with the central part of the slide. I have pretty much covered everything that I could in the slide. So here, when you see DNA is deoxyribonucleic acid, right? So it is going to have nitrogenous bases. This nitrogenous bases can be purines or pyrimidines. Purines, as you can visually see, they have two rings, okay? A six-membered ring and a five-membered ring attached to each other. So, whereas these pyrimidines have only one ring structure. So in this purines, what we classify are adenine and guanine. You can see what is the difference. Maybe in this there is no, in this part there is no difference, but you can see here this bonding is having a NH2 group, whereas here you have a double bond O group. So basically there are a few differences in the composition and the bonding, and that's why we have distinct molecules. But uh, the basic reason why adenine and guanine fall into the purine category is because of similarity of the structure. They are two rings, two membered rings in their structure. Whereas when you talk about the pyrimids, you can again see differences in their composition as well as their bonding structures. So here you can see we have cytosine, uracil, and thymine. I hope by now you will all be aware that cytosine and thymine are present in DNA, whereas this uracil molecule replaces thymine in RNA. Okay, so that is why we have three pyrimids here. In RNA, you have adenine, guanine only. In DNA, also you have these both only. Cytosine is also present in both DNA and RNA, but when it comes to RNA, uracil is present in DNA, thymine is present. That is the key difference. Okay, so going back to this previous question, you can see I have mentioned adenine and guanine as purine, which is right. Thymine and uracil are pyrimidines, that is also right. Cytosine and uracil are also pyrimidines, like cytosine, thymine, and uracil. They are pyrimidines, adenine and guanine are purine. But I have mentioned adenine and thymine as purine, which is wrong. I hope people who are exposed to this for the first time understood whatever I told. If you want me to repeat, just leave a text in the chat box so that I'll repeat it for you again. Okay, this part is done. So what are we going to do with this? Here, this nitrogenous base is attached to a sugar. Okay, here. This R group is what is going to be the differentiating factor between the DNA and RNA. As we all know, DNA stands for deoxyribonucleic acid, whereas RNA stands for ribonucleic acid. So there is going to be a lack of oxy in the DNA. When this R is going to be H, it is going to be a DNA molecule, and when this R is going to be a OH, it is going to be called as a ribonucleic acid or RNA molecule. The nitrogenous base is attached to this ring structure, and when this nitrogenous base and ring structure are attached together, you call them as nucleoside, as mentioned here. Okay, this ring structure and the nitrogenous base together give the nucleoside. When there is going to be attachment of a phosphate group to this, you are going to call it as a nucleotide. So without phosphate, you're going to call it as a nucleoside, and with phosphate, you're going to call it as a nucleotide. So that is the key difference between the molecules that make up a DNA or a RNA. And here, as you can see, this phosphate molecules can be N. That is, N can be 1, 2, and 3. So you will be calling it as AMP, that is adenosine monophosphate, adenosine diphosphate, or a triphosphate, depends on the number of phosphate group that is going to be attached. Okay. Okay. So here, when we come to the rightmost part of the slide, you can see I have given a diagram. This is basically the diagram of a DNA. As you all know, it has two structures. The key difference between the two structures is that you can see 5' end of the DNA is upside in this part of the strand, and it is down in this part of. So before that, let me tell you, this hydrogen bonding is what bonds the two strands. So if I'm imagine this line that projects the hydrogen bond is extended downwards, whatever is in the left side part of it is going to be one strand of the DNA, and the right side part of is assume it as the other strand of the double helix. Okay. So here you can see that they are basically uh, anti-parallel, because 5' and 3' in one direction, and here it is in the opposite direction. So this is what is explained in the second point here. Two strands of the DNA are anti-parallel and connected by hydrogen bonds. Connected by hydrogen bonds is true, but they are not parallel. They are just anti-parallel. We have a 5' and 3' in this direction in this strand, and in this direction in this strand, and this is universal. Like in any DNA, you're going to find any DNA double helix, you're going to find this structure. And one more key feature in this double helix bonding is that always adenine pairs with thymine in a DNA, and cytosine pairs with a guanine. So a purine and a pyrimidine only will bond together, and they will bond using the hydrogen bond. Similarly, there is also the purine pyrimidine bonding is also very specific, where A pairs only with T and G pairs only with C. Okay, so here you can see A pairs with T with two hydrogen bonds, and G pairs with C with three hydrogen bonds. Okay, so the number of bond here is two, and it is three. Uh, Basically, you can understand that breaking of this bond is very easier because it's a double bond. Here it's a triple bond, so breaking is a bit uh, difficult. When you go to a RNA molecule is single stranded. So in RNA molecule, the key difference is that this thiamine will be replaced by uracil, and you can see that uh, this there is a phosphate linkage here. So that is what is going to give you this curved structure or a single strand of DNA bonded to each other. This is basically one nucleotide, okay, because you have a nitrogenous base, you have a deoxyribose sugar, you have a phosphate molecule. So this is basically a nucleotide only. uh, Many such nucleotides are going to get bonded using this phosphodiester bond, and that is why you will be getting a single strand of a DNA. Similarly, when these two single strands are bonded, are complementary to each other and are bonded with hydrogen bond, you will get a double helix of the DNA. Okay, this phenomenon of A pairing with T and G pairing with C is called complementarity. Uh, like when you have a TGCA orientation of one strand of a DNA, the complementary strand is going to be ACGT only. Okay, so that is known as the phenomenon of complementarity between the strands of the DNA. I hope the bonds in the DNA and about this purines and pyrimidines is clear. I'll just wait for one more minute. I'll just pause for a minute so that if you have any doubts or if you want me to explain again, you can just let me know. Like if any of you do not want to post in the chat box, or if you want me to repeat, you can even react. Is there any reaction? Okay, I see a thumbs up there. Let me just for one more time explain this DNA structure. DNA is basically a polynucleotide, as I told you. What is a nucleotide? A nucleotide basically contains a nitrogenous base attached to a deoxyribose sugar in case of DNA, and ribose sugar in case of RNA. So these two components, when they are attached together, they make up the nucleoside. This nitrogenous base and sugar attachment together, when it also bonds with a phosphate, we call it as a nucleotide. So this nucleotide is what is going to make up either DNA or RNA. Based on the sugar moiety, they differ, but both RNA and DNA are polynucleotides. Okay, coming back to this DNA, you can see that we have two nitrogenous base classifications, which are purines and pyrimidines. Purines are two-membered rings, whereas pyrimidines have only one rings. Adenine and guanine fall into the purine category, and pyrimidines fall, cytosine, uracil, and thymine fall under the pyrimidines category. Complementarity is one feature in which one purine, that is this adenine, will form hydrogen bonds only with this thiamine. A, T are only complementary to each other. In this case, guanine is complementary to uh, cytosine. Okay, and here we have this uracil and thiamine, which we can differentiate in RNA and DNA. Uracil is going to be present in RNA, whereas thiamine in the DNA. So what it means, adenine is complementary to uracil in the case of RNA, whereas it is complementary to thiamine in the case of DNA. Okay, so this is what the pairing is going to happen. When you come here to the structure of a DNA is usually a double helix, but as of now, we'll just focus on only one of these strands of the DNA. You can see this part with a phosphate, a sugar, that is a deoxyribose, and the nitrogenous base. This part is known as the nucleotide part. We are going to ignore the yellow color part and see the blue and the green color part. It is called a nucleoside. I think now you are clear with this concept. So this nucleotide is attached to the next nucleotide. This nucleotide has a thiamine nitrogenous base, whereas this has a guanine nitrogenous base. Okay, so you can see that these both are bonded together with a phosphodiester bond. Okay, multiple such nucleotides, when they are bonded to each other with such bond, you call it a polynucleotide or a single strand of a DNA. Similarly, here is one more strand of DNA. You can see this ribose is inverted here. So, this strand is this way 5 prime to 3 prime, whereas the 5 prime end to 3 prime is going to be from this lower end to the upper end in the other complementary strand. Okay, so this is what conferred as the anti-parallelism. Okay, so this is why we call these two strands as anti-parallel to each other. And when these two strands has to be bonded together, we need some kind of interaction between these two. The interaction is that, as we studied, the hydrogen bond formation between the complementary base pairs. You can see adenine complementary to thiamine is bonded with 2 hydrogen bonds, whereas guanine and cytosine are bonded with 3 hydrogen bonds. The bond between guanine and cytosine are uh, is very strong because it is a triple bond, whereas here it is a double bond. Whatever is given in the picture is not just a mere representation, it has its own significance. So I guess it is clear now. If it's clear, can you just give me a thumbs up? Right, okay, thank you for the response. Now we have also seen, so here I'm going to replace T with U. I'm just going to eliminate, there is not going to be any two strands or something, it's just a single strand, and I have U in place of T. I'll be calling it as RNA. Okay, in RNA, as we all know, it is going to have a OH group here. So the sugar is going to be a ribose sugar with which this nitrogenous base is going to be attached. Apart from this, we can classify RNA into different types based on their functions. We have mRNA, rRNA, and tRNA, which are prime importance that all of you should be knowing. So mRNA is basically the messenger RNA. When you are going to go for the transcription process, where this DNA is going to be transcribed into something, it is going to be transcribed into mRNA as it is given here. This is called a messenger RNA because this has the message for the protein synthesis. Okay, this has the prime message, which is going to be converted into a protein, and you all know proteins are of great importance in all of the metabolic activities in any living organism. Okay, so this codes for the proteins. The ribosomal RNA. Ribosomal RNA, as the name suggests, are going to make up ribosomes. Ribosomes will have protein part as well as the RNA part, rRNA part. So their main functionality is that they form the basic structure of the ribosome and catalyze this protein synthesis reaction, which is also known as the translation process. Okay, and tRNA molecules, we also now know that tRNA molecules play a major role in the translation process only. What they essentially do is this is a tRNA molecule. This pink color helical structure that you see is a tRNA molecule, and this green color thing is the amino acid. Okay, when you have a mRNA, you will have a code for a particular amino acid in this mRNA. This tRNA, this part of the tRNA will have the complementary nucleotides for this triplet code that is present here. I'll be explaining this process in detail maybe in the next session. Now I'll just give an overview of this. So it will be complementary. So these both attach, and this amino acid uh, is basically what this code in the messenger RNA confers. So messenger RNA gives the message, ribosomal RNA makes up the ribosome, whereas this amino acid is taken or transferred by the tRNA. Multiple amino acids together are bonded with the peptide bond, and you eventually get a protein out of this process. So that is the role of the three RNAs. Okay, now coming, I'll explain the central dogma in short now, um, and with that maybe we'll just wind up, or maybe I have one more question, so with that we'll wind up. Okay, so here the central dogma is, first it starts with the DNA. The first process in the central dogma, as we all know, is the replication process. Replication, everything, the name itself tells what is that process. Okay, so as it is going to replicate or give two copies of itself, which is essential in cell division process. So when cell division is going to happen, both the daughter cells has to get the genetic information. So we need two strands. Okay, I mean, we need two copies. So this replication process is going to provide us with two copies of the same DNA. And similarly, the second step, or usually the next step, is transcription of the DNA. uh, this is a very important process wherein this uh, DNA will be transcribed into whatever role they have to function. So this will generally contain the information. Now they will be transcribed into their corresponding roles as tRNA, mRNA, rRNA. We now know what is the individual function of this mRNA, rRNA, and tRNA, right? So as I explained a few minutes before, all these RNAs will come together, act with the ribosome unit, and finally the protein is formed. So the simple equation would be DNA gives RNA gives protein. Simple. And when you are going to have a self arrow from DNA to DNA, that is going to be the replication process. And these three steps are the ones which form the central dogma of the molecular biology. So remember, the first step is the replication of DNA, where DNA gives copies of itself. Next, DNA is transcribed to mRNA, and mRNA is translated to proteins. Okay. Okay. So now I'll just move on with the last question for this session. Transport was one of the topics that was covered in week one lectures, so I thought this question is of importance. You can try answering this question. I'll wait for a couple of minutes, and then we'll see the answer in the discussion and wind up the session. Okay, I see pretty less number of participants now. You can just give me some answer which you feel will aptly fit in here, and we'll have a small discussion just for a couple of minutes and then wind up. Any of you would like to answer this question? Okay, if you directly want to see the answer for this question, you can just give me a thumbs up. Okay, okay, B but not sure. Fine, fine. I'll just show the answer for this question now. Okay. The answer for this question is A. Okay. So here you can see facilitated diffusion. So this curve basically tells about the rate of transport plotted over concentration gradient. We all know the transport process usually needs a concentration gradient to move, right? Concentration gradient is the difference in concentration between two sides, so that where the concentration is more from that side, the molecule will move to the other side where the concentration is less. This is called the concentration gradient. Rate of transport is with respect to time, how fast uh, or how does the transport occurs. So you can see one line is linearly increasing, it keeps on increasing, whereas this line is increasing and then it tries to stabilize at one point. So facilitated diffusion usually is something which needs protein or some molecule to facilitate the diffusion process. Similarly, active diffusion is something which will need ATP ATP hydrolysis to drive the diffusion process, whereas passive diffusion is the phenomenon which will not need any carrier or energy for this transport. It will just happen because the concentration gradient is present. Okay, so keeping this context in mind, you can see that when the concentration gradient is going to keep on increasing, that is one side has very high concentration of a molecule and the other side has a very low concentration, you can see that this is this diffusion process rate is going to be increasing. Okay, I hope you understand the nature of this blue curve now. When it is going to be facilitated, so passive diffusion, it is going to increase only, it is not going to have a plateau at one stage, so this option is eliminated. So why facilitated diffusion has this kind of a curve? Coming to that explanation, okay, let me show you here that facilitated diffusion, as I told you, needs some molecules to facilitate the diffusion. So in cell, you cannot have infinitely large number of molecules that is going to facilitate the diffusion. At one point, those molecules are going to be saturated, right? So even though your concentration gradient is increased, your rate will depend on the number of molecules that are present to facilitate the diffusion. Therefore, at one point, this rate of transport is going to saturate and become a plateau. That is why this curve increases and eventually saturates. This saturation happens because there is limitation in the number of molecules that facilitate the diffusion process. Okay, that is what is given in the explanation of the slide also. I hope it is clear. If you want me to repeat, give me a thumbs up so that I'll repeat it. Okay. Um, See, let me go back to this picture only. As I told, concentration gradient is something that is going to drive the transport process because it means that there is a difference in concentration between two parts. Okay, in one side the concentration is going to be very high, whereas in the other side the concentration is going to be lesser. So eventually from higher side the molecules will move to the lower side to bring in a state of equilibrium. Okay, rate of transport is something, rate with respect to time, how the transport is going to be. So we are going to plot it as a graph. When we are going to plot it, we see that along this way the concentration gradient is going to increase. So along this way the rate of transport is going to increase from bottom to top. Okay, so when you are going to plot it, when the concentration gradient is increasing, rate of transport is also increasing. So this is very linear relationship and it is increasing linearly. Here you don't use any molecules which facilitate this process or which make this process happen. Whereas, as the name suggests, in facilitated diffusion, you will have molecules which are going to facilitate the diffusion process. So, how much ever high the concentration gradient is, that is, there are few helper molecules that are going to help these molecules to get transferred. Okay, even though there is a very high concentration on one side and low on other side, the helper should be sufficient. Let me tell in a very layman term, not very scientific. 1,000 people, out of 1,000 people, 500 people have to travel from one side to another, and we have only 250 people who can help them do this transition from one side to another. Then, even though there are 500 people to be transported, only 250 will be transporting. Initially, when there are one people, it will be here, two people, three people. Once this 250 people are reached, or 250 helpers are filled with some work, you will not eventually have other free molecules to facilitate. Okay, so that is why this will increase for a certain period, and once the facilitated molecules are all saturated, it is going to saturate at this point, and that is why the graph increases and then saturates. I hope it is clear. Right. Okay. So this was the last question for the day, and I'll just ask for a few suggestions. Let us continue the sessions like this for the upcoming weeks also, or do you have any suggestions? You can just let me know in the chat box, or okay, okay.